Welcome to the McMaster University Demystifying Medicine series. This video will provide a brief overview of Dr. Arias' work on the polarization of cells in the liver. This is a neglected topic, only mentioned in one textbook. Let's get started with some basic information about the liver and how it works. The liver is the largest internal organ of the body, accounting for 2 to 3% of your entire body weight. It is located beneath the diaphragm and spans across the abdominal cavity, with most of the mass located on the right side. The liver is protected by the rib cage. The liver is a complex organ with many different levels of organization. There are 4 distinct lobes of the liver. The right lobe is much larger than the left lobe. The caudate and quadrate lobes are smaller and located on the posterior side, or the back, of the liver. The liver can be further divided into 8 functional segments. Each of these segments consists of thousands of lobules. The lobule is the functional unit of the liver. A vein, artery, and bile duct on the outside of the lobule make up the portal triad. The vessel in the center of the lobule is called the central vein. Millions of cells are found in each lobule. The hepatocytes account for 70% of the cells in the liver. There are also a number of immune cells present that function to protect the body against foreign organisms. Blood flows from the portal vein and portal artery through tiny tubes called sinusoids, and then into the central vein, which eventually leads back to the heart. In fact, 40% of the blood returning to the heart is actually coming directly from the liver. Bile produced by the hepatocytes are collected in the bile canaliculi, and then travel to either the intestines or the gallbladder through the bile duct of the portal triad. The liver simultaneously acts as a storage facility, manufacturing unit, and processing plant. Each of these functions are so important that without the liver, our bodies would simply stop working. One of the liver's main functions is to filter the body's blood. Blood that is received from the intestines contains fats, carbohydrates, vitamins, and other nutrients dissolved in it from the food you've consumed. All of these are processed in different ways. In the case of carbohydrates, the liver breaks them down and converts them into sugars for the body to use as energy. When the body has leftover sugars, the liver keeps them in storage for future use. The blood flowing into the liver also contains toxins that the body can't use. When the liver spots a toxic or useless substance, it either isolates and excretes it in the bile, or it detoxifies it, converting it into a product that won't harm the body. This organ also makes everything from various blood plasma proteins that form blood clots to the cholesterol that is needed to make hormones. But one of its most vital products is bile, a substance that breaks down fat, destroys microbes, and neutralizes stomach acid. When discussing the function of hepatocytes, cell polarity is an important topic of consideration. Cell polarity refers to the difference in the structure, function, proteins, and biochemical properties of different cell surfaces that arise from the specific arrangements of cell components. This concept is explored in a paper by Gisson and Arias, published in 2015. Along with hepatocytes, the polarization of cells is commonly seen in neurons and epithelial cells, which have a basal and apical surface. These surfaces differ mainly by the presence of different structural components designed to carry out specific functions. For example, little hairs called microvilli are present only on the apical surface of hepatocytes. They significantly increase the surface area of the cells, allowing for increased cell productivity. The basal surface has specialized junction proteins, which allow it to adhere to the basement membrane. Polarization is vital to the function of cells, and a discrepancy in polarization can lead to abnormalities and disease. Something unique about hepatocytes is their multipolar organization. This means that they have multiple basal and apical surfaces. This allows for the exchange of macromolecules, a vital function for the liver. This video has only begun to outline the fascinations of the liver. Research on hepatocyte polarization is limited, and there are many more questions that must be answered before we can fully grasp this concept.